Okay, hello everybody, and welcome to this session on the Radiation Effects Research Foundation. My name is Elizabeth Cardis. I'm the head of the radiation program at the Barcelona Institute for Global Health in Barcelona, Spain. And it's a great honor for me to chair this session since I started my career in radiation epidemiology at the Radiation Effects Research Foundation in Hiroshima many years ago. So thank you very much to our colleagues from Hiroshima for staying up or staying at work late to allow people from Asia and Europe at least to attend. So I don't want to waste much time, just a couple of announcements. So the presentations will be 15 minutes each. We will allow 1 to 2 minutes maximum of pressing questions right after the presentation, so we can have about 10 minutes at the end to have a general discussion. Please put your questions in the chat and I will read them. Make sure that your microphones are muted. And I think that's it. So I'd like to start with Dr. Ozasa, who will give us a brief introduction on RERF and talk about cancer incidence in the lifespan study. Dr. Ozasa.
Thank you, Dr. Elizabeth Cardis. Hello, everyone. I'm Kotaro Ozasa, and I will share my slide. Here. Okay. Today, I'd like to introduce the RERF briefly and talk about the radiation risk of cancer incidence in the lifespan study of atomic bomb survivors. Hiroshima and Nagasaki were attacked by atomic bombings in August in 1945. Around 40% of population of Hiroshima city and 30% in Nagasaki died within a year. Cities were destroyed by blast and fire by the bombings, and people suffered from physical and burn injuries and acute radiation syndrome. Later, leukemia increased rapidly, and other cancers increased gradually among the survivors. ABCC was established in Hiroshima and Nagasaki in 1947 and 48, respectively. In the early period, physical and radiological injuries were investigated medically. Then, late health effects of radiation and its biological mechanisms, including genetic effects, have been investigated based on the fixed cohorts and laboratory and animal studies. Dosimetry system of atomic bomb radiation has been developed and individual radiation doses were estimated. The estimates continue to be updated as new technologies become available. In 1975, ABCC, the American organization, was reorganized to RERF, a public interest incorporated foundation under Japanese law, co-funded by the US Department of Energy and the Japanese Ministry of Health, Labor and Welfare. Activities have continued uninterrupted through this transition period. This photo shows Hiroshima City now. RERF is located on the top of Sugiyama Hill. Here are the hypocenter of the bombings, the atomic bomb dome, and Peace Memorial Park and Museum. In Nagasaki, RERF has another laboratory. This slide shows the current organization and activities of RERF. Our department is conducting the epidemiological follow-up of the cohorts of survivors in neutral-exposed persons and children of survivors. The Department of Clinical Studies is conducting health examination programs for sub-cohorts in Hiroshima and Nagasaki laboratories. The Department of Molecular Biosciences is involved in the investigations of transgenerational radiation effects and laboratory and animal studies for radiation carcinogenesis and other pathogenesis. The Department of Statistics is engaged in statistical analysis and dosimetry system. The Department of Information and Technology manages our research database. The Biosample Research Center stores, inventories, and manages the biosamples obtained from our study subjects. The Research Resource Center is a new center currently under development that will integrate our research information to improve our ability to do research. I'd like to introduce some programs for international training of young investigators and others. Before COVID-19, we obtained a yearly allowance from the Japanese government to hold 2-week programs to provide epidemiological and statistical education for a few overseas participants. When the pandemic passes, we expect that this program will resume. The Department of Statistics is hosting, through the Japan Society for the Promotion of Science, JSPS fellowship mechanisms, postdoctoral researchers in the areas of statistics, biostatistics, bioinformatics, genetic epidemiology, or related fields who are interested in pursuing methodological research in areas relevant to the study of the health effects of ionizing radiation. If you are interested in these programs, please ask on the contact page of RER homepage. Another is the partnership program based on a contract with other institutes. We had one previous example with the University of Washington for education of postgraduate students based on the budget from the US government. We have also allowed students from various universities in Japan and overseas to do internships in various departments. But this generally requires the students to have their own sources of funding. Of course, these activities are currently halted under the pandemic, but we hope they will resume in the future. From this slide, I'll introduce the results of radiation risk analysis of cancer incidence in the LSS from 1958 to 2009 using updated individual doses DS02R1. And this is the first time that radiation risk was adjusted for potential confounding factors such as lifestyle and other factors, in addition to the traditional confounding factors of age at bombing, attained age, city at bombed, and so on. Also, age at bombing and attained age were included in effect modifiers if significant, as usual in the previous studies. The risk estimates for age at bombings of 30 and attained age of 17 years is shown as representative values. Effect modification by lifestyle or other factors were particularly shown in lung cancer by smoking and in female breast and uterine corpus cancers by reproductive factors. So as a total, 22,000 solid cancers occurred among about 100 persons in the LSS. For all solid cancer, ERR for radiation was around 0.5, almost similar to those estimated in previous studies. but the most fitted dose response shape was linear quadratic in males and linear in females. The other important point was that radiation risk was not confounded by smoking almost at all, although smoking is a potential strong confounding factor for cancer. Radiation risk estimates over the whole dose range was determined mostly by the data at 1 gray or higher. People who were exposed to 1 gray or higher red dots in these maps distributed in the narrow areas near the hypocenter, and they were exposed to radiation non-selectively. That is, for example, there is no association between smoking and radiation exposure. So there is no substantial bias or confounding by other factors for radiation risk estimates. On the other hand, persons who are exposed to low dose radiation distributed wide area in Hiroshima and Nagasaki rural areas. So there are many factors that we have to consider, especially for geospatial factors related to distance from hypocenter when we evaluate low dose radiation risks, because distance from hypocenter is strongly related to individual doses. There are other factors that we have to consider, but I don't touch details today. Excuse me. Next, I'll talk about individual sites. Risk of cancer of the digestive system increased similarly to the previous studies. Colon showed an increased risk as well, and especially in this paper, proximal colon showed a higher risk compared to distal colon at the first time in this paper. Rectum didn't show any increased risk similarly to the previous studies. Liver showed an increased risk as well. Pancreas showed an increased risk probably at the first time in this paper. Lung showed an increased risk as in the previous studies, and the positive interaction between radiation and smoking was also observed. Female breast cancer and uterine corpus cancer indicated increased risks, but uterine cervical cancer didn't show any increased risk. Here, radiation risk of breast cancer and corpus cancer was strongly associated with activity of secondary sexual development. Radiation risk of breast cancer was higher in women who had earlier menarche, and the radiation risk was the highest when they were exposed to radiation around at age of menarche. For uterine corpus cancer, the risk was the highest and significant when women were exposed at age just before the menarche. This pattern of sensitivity to radiation during the puberty period is thought to be associated with the development of mammary gland and uterine endothelia, that is, activity of tissue stem cells of those organs. The next is cancer in the urinary tract and bladder. They had an increased risk similarly to the previous studies. Ovaries showed an increased tendency in risk, but not significant. Central nervous system showed an increased risk as in the previous studies. Finally, this is the first time that the prostate cancer showed an increased risk of radiation. Actually, about 2/3 of these cases were detected after 1995, since incidence of prostate cancer has rapidly increased in Japan recently. Therefore, in conclusion, there are some important findings in the series of analysis of individual cancer incidence due to increased number of outcomes, adjustment for relevant factors, a recent change in dominant cancers in Japanese population, update of individual dose estimates, and so on. Thank you very much for your attention.
Thank you very much, Dr. Ozasa, and thanks for respecting the time. Do we have any specific questions to this presentation? Just maybe just one comment. I was very interested in what you said about the potential for confounding, so that the people with more than one gray, because they were very close to the hypocenter, it was more or less a natural experiment, so that there's less selection and so on. But people with lower doses, there was more potential for confounding by many different factors. Have you done any analysis just focusing on the low dose range to see whether the effect of the confounders might be greater?
Oh, you mean the low dose fine? So
I mean, because obviously for example, smoking for all cancers, the effect is very small, as we saw when you take the entire data set. But if you restrict to lower doses, say, I don't know, below 200 milligray, for example, is the effect of confounders stronger?
Yeah. For example, at that time, the distant survivors were located in the rural area, so the lifestyle factors are different from the urban residents, proximal survivors. So such kinds of difference may be confounding factors.
All right, let's leave time for the discussion at the end. Thank you very much. The next speaker is Dr. Alina Brenner. Alina?
Hello, everyone. Let me pull my slides. Can you see the slides?
Yes, thank you.
All right. Again, thank you for the opportunity to share with you today the results of comparative dose response analysis for all solid cancer in cancer mortality and incidence LSS data. Sorry. Okay. In the lifespan study and other studies with whole body homogeneous exposure to ionizing radiation, solid cancers are often combined to increase statistical power and precision of radiation risk estimates, with the assumptions that there is homogeneity in the background rates and those response estimates across different sites. In early LSS studies, there was no evidence against linearity for all solid cancer mortality or incidence data. However, in the latest mortality report, there was evidence for the upward curvature in sex average excess relative risk below 2 gray. Also, in the most recent incidence reports that Dr. Ozasa mentioned in his presentation, we found evidence of the upward curvature in excess relative risk among males, but not females. Moreover, in a follow study of cancer incidence by Colon and colleagues, they demonstrated that by excluding individual cancer sites from all solid cancer incidence, there was significant variation in the magnitude of dose response curvature for the remaining sites, not only among males, but as well as females. The sources of curvature in mortality data are investigated to a less extent than in cancer incidence data. Also, it remains not entirely clear whether mortality and incidence data provide consistent evidence for the upward curvature in either sex. And part of this is due to the differences in data and how the data were analyzed. I summarized these differences in the following table, and you can see that the follow-up in two analyses was different. The northern city at the time of the bombings, subjects were treated differently, doses were different, and importantly, the statistical models that were applied to the analysis of solid cancer mortality and incidence data were quite different, and differences involved both different ways of modeling baseline rates as well as dose response. Another difference that is important to keep in mind and is specific to LSS data, that incidence data is ascertained via regional cancer registers in Hiroshima and Nagasaki, while mortality data are available through nationwide monitoring of death certificates. And consequently, the person years for the analysis in incidence data are corrected for migration probability into and out of the catchment area, and person years for mortality are unadjusted. So the objective of the current analysis was to evaluate sex-specific dose response shape for all solid cancer in LSS mortality and incident data, applying similar methods and statistical models. And secondary objective was to explore factors contributing to all cancer dose response curvature. As before, analyses are based on Poisson regression methods for group survival data. For the same number of people and follow-up, there were nearly 3.8 million person years in mortality data and about 15,000 deaths from solid cancer. The number of person years in the incidence data was slightly fewer, but there was substantially more cases of solid cancer. In fact, 46% more, underscoring that incident analyses are more powerful than mortality. We fitted sex-specific linear and linear quadratic excess relative risk model of the same form as in Grant et al. Most recent analysis of cancer incidence. And as Dr. Ozasa mentioned, both analyses were adjusted for smoking. In some analyses, we allowed the dose response parameters to vary by calendar period or age of exposure. In addition, for selected cancer sites, we conducted joint endpoint analysis to evaluate the effects of site-specific heterogeneity on the magnitude of dose response curvature for the combined outcome. The main results are summarized in the following table. The evidence of dose response curvature was found for males, both in mortality data and incidence data of the entire range of doses, as well as below 2 gray. The difference was for females, as we found the evidence of upward curvature among females for mortality, but not incidence. And the results below 2 gray were similar. I would like to add that similar findings concerning mortality were in additional analysis in which deaths that occurred outside of Hiroshima and Nagasaki were excluded, and present years adjusted for probability of migration as in incidence data. Also, the results were similar when we used full follow-up for mortality for 1950 and 2009. In this slide, you can appreciate the differences between mortality and incidence data in the visual form. The 2 upper panels represent the fitted dose response for females, and the 2 low panels are for males. The mortality data are on the left and incidence data on the right. Once again, I would like to repeat that the main difference between mortality and incidence occurred in females. The dose response in females, mortality data followed linear quadratic function, while the dose response in incident data was strictly linear. The dose response for males was essentially identical in mortality and incidence data, and both was well described by a linear quadratic function. So, what are the differences that potentially could contribute to difference in sex-specific dose response shape in mortality and incidence data? We ruled out various differences in type of data and dose response models, use of different dose estimates, etc. So what it seems to be is that the differences are related to data themselves. In other words, to the fact that mortality is based on deaths and incidence on newly diagnosed cases. And on this slide, you can see comparative distribution of mortality and incidence data by site. Due to the good survival, the fraction of cancers with favorable prognosis, such as breast, thyroid, brain, prostate, in mortality data is substantially lower than in incidence data. And this difference is particularly strong for females because breast, thyroid cancer, benign brain tumors are substantially higher in females than in males' incidence rates. Moreover, these cancer sites demonstrate very strong and linear dose response in incidence data. So their deficit in mortality data could reveal non-linear dose response if it exists. Now I would like to switch to the analysis of curvature by calendar time. So in this analysis, we use the mortality and incident data of entire follow-up, but allowed sex-specific dose response parameters to vary by time. And we split time in 2 calendar periods, splitting number of cases both in mortality and incidence approximately by half. What you can see from this summary table that we found that nearly all evidence in mortality curvature for both males and females was coming from recent calendar period, and there was no evidence of curvature in early calendar period. The pattern of incidence curvature was different. The evidence for male curvature was present both in late and early period, and there was no evidence for female curvature in either period. I have to say that in LSS, there is a correlation between time since exposure and aged exposure. And recent data are strongly influenced by data from those who were young at the time of exposure. We also conducted analysis, but I do not have time. I just would like to mention that these findings are interesting exploratory analysis, in which those response parameters were allowed to vary jointly by calendar time and aged exposure. And then in this analysis, one consistent pattern emerged. That is, evidence of curvature was present among both males, females in cancer mortality and incidence data for those who were less than 20 at the time of exposure and who went through a recent calendar period. Trying to understand where the temporal differences come from, we focused on mortality data because mortality showed very clear pattern by time since exposure, both in males and females. And look at the distribution of mortality by cancer site. You can see here the data that are presented for females and males. The gray bars are contribution and fraction of cases that occurred during early calendar period, and the black bars are for most recent period. And you can see that for the same age at death, the fraction of stomach cancers in all solid cancer mortality was reduced drastically, both in females and males. At the same time, the fraction of deaths due to lung, liver, colon increased, both in females, and the same pattern was seen in males. Collectively, these sites accounted for 68% of all deaths in cancer mortality and nearly 53% of deaths among for males, and for females. So I selected these sites in order to conduct joint analysis of 4 sites and explore how unaccounted heterogeneity and background rates could impact the estimates of curvature. So first, we conducted standard analysis when all parameters in the background were common to 4 sites. And these are the estimates for males and females. The evidence of curvature in males for these 4 sites was somewhat weaker than for all solid cancers. In females, it was stronger, but it was following the overall patterns that was seen for all solid cancer mortality. The big surprise was that by allowing for substantial site-specific heterogeneity in background traits, we didn't see much effect on the dose response curvature for the combined outcome. It was rather surprising. We then looked at the dose response shape for individual cancer sites.
1 minute.
And 1 minute. Oh, I'm sorry. Okay. Yes. And we found that there was an evidence of heterogeneity in dose response shape. In that colon cancer mortality was best described by pure quadratic model. So it was another surprise. Now, trying to summarize different pieces together, I want to say that under similar analytical methods and rate models, there was consistent evidence of a modest upward curvature in all solid cancer dose response among males in mortality and incidence data. By contrast, the upward dose response curvature among females was specific to cancer mortality, and there was no evidence of departure from linearity in incidence data. Nearly all evidence of curvature in mortality data appeared for both sexes after 1988. And there was little evidence that curvature estimates varied by calendar time in incidence data. But as I said before, the hints were that both time and aged exposure are important factors contributing to evidence of curvature. Our analysis also suggests that the evidence of dose response curvature appears to depend on the composition of cancer cases. And both types of heterogeneity in terms of the heterogeneity and background rates and dose response shape are also important in order to understand where the curvature coming from in all solid cancer dose response. Finally, with 84% of survivors who were who are alive and were under age of 20 years at the time of the bombing, it's very important to continue the follow-up and to see whether the emerging patterns of curvature would persist in the future or get stronger because of data from young survivors. And in closing, I would like to thank and acknowledge my collaborators for their helpful advice and input. And thank you for your attention. Sorry if I went over time.
Thank you, Alina. Sorry. In the interest of time, I propose we move directly to Dr. Sugiyama's presentation, and then we can take any specific questions in the general discussion. Thank you. Dr. Sugiyama.
Can you see my slide?
Yes, thank you. Perfect.
Okay. Shall I start? So thank you, Dr. Cardis and Dr. Costi, to organize this seminar. So today I'm very honored to be here to introduce the updated results of the mortality of individuals exposed to atomic bomb radiation in utero in Hiroshima and Nagasaki. And this paper was published in European Journal of Epidemiology last year. So I will start with the introduction of the previous results and the in utero cohort, followed up by the RERF. So RERF reported the antenatal radiation, atomic bomb radiation, increased mortality risk for solid cancers, so followed up 1992. So we extended the follow-up up to 2012. While chronic radiation did not increase the cancer mortality risk in Southern Ural cohort members and the other members. So in the RERF in utero cohort were originally comprised of two cohorts. The first cohort was derived from clinical study program, and the other was derived from the mortality study program. The clinical cohort members were alive as of 1st of October in 1950 and lived in Hiroshima and Nagasaki. And the members of this cohort were finally fixed in 1959, and they were followed up clinically until they reached 19 years old. And Otake and Shur found that the radiation dose were associated with the prevalence of individuals with small head size and with mental retardation. And the other cohort members of the mortality program were selected based on these several sources. The mortality study program revealed the distribution of types of cause of death among the cohort members during early period. And Kato and Shar colleagues also conducted a male-based socioeconomic survey in 1964. So they found the high frequency of individuals with small, low birth weight and high frequency of individuals who lost one or both parents in the proximal area. So at that time, the radiation dose was not estimated yet. So now we hypothesized the relationship between atomic bombings and mortality. So usually, age, sex, city, and other variables are important factors for describing mortality in a certain population. Now, we would like to evaluate the effect of antenatal radiation exposure from the atomic bomb on mortality. So based on the evidence from clinical studies and the socioeconomic survey, the prevalence of individuals with a small head size, low birth weight, and individuals with small head size, prevalence of low birth weight, and loss of parents were all associated with distance home hypocenter. So they might be affected by atomic bomb radiation, and also they might affect mortality. So now we assume these factors are potential mediators between radiation and mortality. So however, we need to keep in mind several factors other than radiation shown here as malnutrition and poverty. So during pregnancy or after bombing to birth might affect these three factors. So also these unmeasured factors might affect childhood or adulthood mortality. The aim of this study is to examine mortality of in utero exposed to atomic bomb radiation, so taking into account these potential mediators. So in the next few slides, I will introduce the method of this study. The number of in utero cohort members was 3,638. The subjects were exposed to atomic bomb radiation in mother's womb who were born after the bombing through May 31st in 1946. So these figures show each mother's location at the time of bombings in Hiroshima and Nagasaki. So with red crosses showing the hypocenters. So we used DSO2 revision 1, mother's weighted absorbed uterine dose in this study. So because unfortunately the RERF has not estimated the fetus dose so far. So after excluding ineligible cases, the final number of the subjects for analysis was 2,463. The follow-up period was basically from 1950 to 2012. And the vital status were ascertained by the Japanese family registry system, and the underlying cause of death as an outcome, so were ascertained by this certificate. The first part of the statistical analysis was to examine the association between radiation dose and potential mediators. And the logistic regression and multinomial logistic regression were used for each response. The response variables were birth weight, head size, and the parent survival status as a surrogate of complexity of family status. The main risk factor was radiation dose. The odds ratio or relative odds ratio at 1 gray for each response was estimated. So radiation dose was adjusted for these factors. So then the association between antenatal radiation exposure and mortality was examined. So outcomes of interest were solid cancer death, non-cancer disease death, and external cause of death. And the Poisson regression was used to estimate the radiation-associated ERR using individual person years data. The basic ERR model is explained here. So lambda 0 shows the background rate, rho shows the ERR function. So then potential mediators were added one by one into the background model. I checked for any changes in the radiation-associated ERR. Okay, so let's move on to the results. The characteristic of the subject are shown here. The potential proportion of males and females was almost the same. The proportion of Hiroshima members was 83%, which was much higher than Nagasaki. The maternal uterine dose distribution was very skewed. So about half of the subjects were exposed to less than 5 milligray. So we observed 339 all-cause deaths as a whole. The number of solid cancer deaths was 137, including one childhood cancer death. And that of non-cancer disease deaths was 134. So we observed 56 external cause of death. And males are predominant for all types. So this figure shows the association between atomic bomb radiation and potential mediators. The low birth weight, small head size, and loss of father were positively associated with atomic bomb radiation. The association between radiation dose and loss of mother was not so strong. Okay, so I'm going to talk about the radiation-associated ERRs and the relative risk of potential mediators for solid cancer death firstly. The upper radiation ERRs are shown without adjustment for potential mediators, while the lower ERR are adjusted for all these variables. The elevated radiation ERR for females was estimated to be 2.24, but the ERR for males was not elevated. After adjustment for potential mediators, the ERR slightly increased, and none of the potential mediators were associated with solid cancer mortality. Then we examined adulthood solid cancer mortality, and the result was the same. For non-cancer disease deaths, so elevated radiation ERRs were observed for both sexes. So after adjusting for potential mediators, the male ERR was dramatically attenuated from 1.22 to 0.39, 68% decrease. The female ERR was attenuated from 2.86 to 1.48, so dropping by about half. The low birth weight and small head size were positively associated with non-cancer disease mortality. The loss of father was suggestively associated with non-cancer disease mortality. For adulthood mortality, the ERR for females was elevated at 2.23, but not for males. And after adjusting for potential mediators, the ERR for both sexes were attenuated. And small head size was positively associated with non-cancer disease mortality, while low birth weight and loss of father were suggestively associated with non-cancer disease mortality. For external cause of death, the ERR for females was elevated at 2.57, but not for males. So after adjusting for potential mediators, the ERR was attenuated. And loss of mother was associated with external cause of death. For adulthood mortality, the confidence intervals of ERR for both sexes become wide, and the lower limit was less than 0. The females with small head size showed a higher risk of adulthood mortality of external causes of death. Okay, all right, here we are at discussion and conclusion. So we observed 339 deaths from 1950 to 2012. So radiation-associated ERR for solid cancer deaths was increased in females. The ERR for non-cancer disease deaths was increased in both males and females. The ERR for external cause of death was increased in females. So radiation dose was positively associated with potential mediators, but not mother's survival status. So in order for a child to be born, the mother must survive. The low birth weight was associated with radiation dose. So however, the Chernobyl study did not find a relationship between the birth weight and in utero exposure to radioactive iodine 131 at fetal thyroid dose. And several risk factors of low birth weight have been reported, such as food shortage, mental stress, the disruption of infrastructure, lack of access to medical services, so particularly during armed conflict. So the influence of physical injury of mothers and the social factors that were not measured in this study, so need to be considered for the effect of atomic bombings. The low birth weight, loss of father, and small head size for female increased the mortality risk for non-cancer diseases. And the loss of a mother and small head size for female increased the mortality risk for external causes. So as a limitation of this study, we used the maternal uterine dose as a proxy measure. So because fetal radiation dose is by gestational age at the time of bombing could not estimate it. The lifestyle and socioeconomic status in adult life were not obtained. The subjects have been followed since 1950 or later. So in conclusion, so we found that the atomic bomb radiation was associated with mortality from solid cancer for females, non-cancer disease for both sexes, and external cause for females. So then we also found that the atomic bomb radiation is associated with low birth weight, small head size, and loss of father, so which affected non-cancer mortality, but not solid cancer. So therefore, antenatal radiation is a consistent risk factor for solid cancer mortality in females. So there is evidence of mediation effect between atomic bomb radiation and mortality from non-cancer disease and external cause. For pregnant women and in utero fetuses, so several unmeasured factors related to the atomic bombings could have caused low birth weight and poor survival status of parents. So careful interpretation is needed regarding the relationship between atomic bomb radiation and these potential mediators. So this study was conducted with these co-investigators. Thank you for your attention. I'm very happy to take your question and comment.
Thank you very much, Dr. Sugiyama. Very nice presentation. Do we have any specific questions to this presentation?
I have some doubts as to whether the mediation analysis is actually the right thing to do here, because you're effectively you're adjusting for something in the background which is related to dose. So I wonder whether you may be over adjusting. I mean, and I find it interesting that there was effects on non-cancer, but not on cancer. I mean, perhaps could you also indicate which types of non-cancer mortality you had in your data?
So in the paper table 4, I show the number of the deaths by cause of death, more finely classification. So you can check the number of what kind of the diseases are most frequent or not. And so, but in the table 4, non-cancer disease includes the circulatory disease and the respiratory disease, digestive disease, like that. So, but I did not estimate the ERR for each precise cause of death.
You can't do that.
Yeah.
Aren't enough case deaths, obviously, but yeah.
Right, it's a small number of the cases.
Yeah, I agree with Mark about whether these are mediators, and I'm not quite sure that they would be. I mean, I don't offhand intuitively see any mechanism for mediation, but you know, these would be possibly more indicators of doses. Because how do you interpret this mediation effect?
So I partially agree with the over adjusting to use this method. So, but the radiation, atomic bombings include radiation of many factors or affect to the mortality. So I just picked up any potential mediators, and I want to interpret the atomic bombings, how atomic bombings affect the mortality in individuals exposed in utero subject.
We have 4 minutes left, so I think we should probably have a more general discussion. There was a question by Francisco Boquicchio about thresholds, but I think that's been answered. There was a question by Robert Hayes about saying that the graphs show that as dose approaches 0, there was no negative ERR values. And just from statistical variation, wouldn't we expect to have some negative ones? Anybody has an answer?
Yes, probably there is a variation from positive to negative, but I don't remember exactly how much it was.
Okay. Any other questions? General questions about the presentations, or questions about possible collaborations or stays at RERF?
I had a question to Alina about the analysis, and you'd expect that there would be a lag between incidence and mortality. So, I mean, did you consider looking at sort of, well, in other words, cutting off the incidence analysis a few years early, so before 2009, to get the so that you're comparing to some extent like with like, and I can't imagine it will have much effect from what you were saying, but it might be worth just looking at, just moving the follow-up back, as it were, for the incidence data.
Mark, I see your point. Yes, in fact, because always there is a lag between incidence and mortality, and it's stronger for cancers with good survival than cancers with short survival. We did not look this way in the data, whether there is, in other words, whether there is continuous increase rather than binary analysis. This is what you are suggesting, looking not only like in 2 periods, by rather multiple, trying to see the trend over time of entire, whether there is monotonic increase, let's say.
Well, I would propose just doing a simple analysis where, for example, you cut off, say, 2 or 5 years of the incidence follow-up at the right at the end, and just see what effect that has. I mean, I can't imagine it's going to have much effect, but I suppose it'd be worth looking at.
Okay, certainly we can look at this. No, I did not assume any lag. Yeah.
Is it also, is the analysis you did adjusting for essentially for the difference in background, is that quite similar to the what's presented in John Cologne's paper of a few years ago, or is it a different type of analysis?
Mark, in John Cologne's paper, he looked at cancer incidence only. He did not.
I appreciate, yes, but yeah,
yes,
but it's just the type of analysis that I'm interested in, not the data, as it were.
The statistical approach was similar, but if you recall, in his analysis, he looked at the linear dose response. It also depends. There were two papers by John Cologne, okay? One which looked at the magnitude of curvatures over the same follow-up by excluding one site at a time. Second, he looked at the estimates of linear ERR over calendar period of time. So, I'm sorry, what analysis are you referring to?
I'm sorry, we're running out of time here, so we should not get into too much detail.
I'll follow up later, Alina. I'll follow up offline.
Yeah, okay, so, but the analysis was similar to the way he did, only looking at the linear quadratic form as opposed to linear over time.
Is there, I mean, we're over time already now. Is there any last pressing question?
May I ask?
Yeah, Christian.
I'm Christian Strafe from Germany. Which cancers did you see, especially with the females? I think this remarkable difference between females and males. Can you say anything about the entities of cancers which you observed?
So which are the types of cancers that would justify this big difference you see?
Yes.
Is it question to me or is
Yes.
it question to some? Okay, difference in terms of the, as I showed at the beginning of the presentation, the structure of cancer mortality and incidence for females is more different than among males. And this is due to the fact that in females, incidence data, there is much stronger contribution of breast cancer, thyroid cancer, benign meningioma, and other gynecological cancers. So this is the main difference. The biggest difference for males is prostate cancer. And the point is that these cancers have good survival. Therefore, their representation in mortality data is limited.
Is this also the case with the in utero exposures?
The numbers of cases in the in utero exposures in utero study probably prevent any such study, no? So the question about to you, Dr. Sugiyama, was whether you also see the same differences between males and females in the in utero study.
Yes, that's the point. And is there any explanation?
Sorry, I tried to find out why there is a difference between the male and the females. So, but the number of the cases are very small, so it's difficult to interpret it. Sorry.
Thank you, Alina. I think you've done a really nice job of looking at the drivers of these differences in curvature between males and females. And also over time, it's very interesting. Any more questions? We are now 5 minutes over time. No, well, I'd like to thank our speakers and to thank all the participants and to thank Professor Niwa, who's also here with us from Hiroshima today. I think it's been a very, very interesting session. And I'd like to remind you, Rania posted this in the chat, that the next ISoRED webinar will be on February 17th. And we'll hear from our colleagues at the UK Health Security Agency, which is the former Public Health England. So thank you very much. And I presume that if you have any further questions, you can send emails directly to our speakers. Thank you and goodbye.
Bye-bye. Thank you.
Thank you.